Hello, grade 12 students. In this video, we're going to talk about the midterm exam review worksheet. So, question number one, PKU. Phenylketonuria is a metabolic disease of genetic origin that affects 1 over 10,000 child worldwide. So, the frequency worldwide it's 1 over 10,000. It's characterized by the accumulation of the phenylpyruvate in blood. This product is negatively affecting the neurons, especially in children younger than 10 years old, resulting in severe neurological disorders. Normally, the amino acid phenylalanine is metabolized by the enzymes called PAH, phenylalanine hydroxylase, which need DHPB as a cofactor. The most frequent reason for the appearance of this disease is a malfunctioning enzyme, PAH. The excess phenylalanine will be transformed into phenylpyruvate. Prenatal diagnosis may help to detect this genetic defect. A diet which is poor in phenylalanine sources like the egg white, soy protein isolate, seaweed, and peanuts, etc., during the first 10 years of life may help to save the child from the expected troubles. What do these suggestions provide regarding the dominance or the recessiveness of the disease? So, the appearance of this disease, as they said, is mainly due to inactive or absent PAH. So, the presence of one allele coding for normal, which is the active or the functional, will lead to normal. As such, we can know that affected person which should have two affected alleles or two defective alleles. So the mutated allele or the affected allele, it's recessive with respect to the normal dominant allele. And this is mentioned only in the text. Part 2. Statistics done on isolated community allows to detect 20 heterozygous children out of 80. So 800, sorry. So 20 over 800, this is the frequency of being hybrid or heterozygous in the community. So 20 over 800, it will be 1 over 40 to be hybrid or to be heterozygous, as we know. So here in the community. So they said, propose a hypothesis that explains the difference between the rate and the world rate. This can be explained like a hypothesis colon. The marriage of the relatives can lead to the increase in the risk of having PKU children. And also, what is the risk to having affected child in this community? First of all, both parents should be heterozygous, so 1 over 40 for the father times 1 over 2 times 1 over 40 times 1 over 2. So it will be 1 over 6,400. 40 times 40 times 2 times 2. Or you can say frequency of the father to be heterozygous times frequency to the mother to be heterozygous times half, which is times 1 over 4. The following pedigree is that of this closed community, of which some members are affected by the disease. So here, the two sisters, they consulted a doctor. So 3-3 and 3-6, they consulted a doctor and to evaluate the risk for them for having an affected child. So what is the risk of having an affected child or affected fetus, like the case in 4-2 and 4-3? So they consulted a doctor. Let's see what happened. They say identify the chromosomal localization of the studied gene. According to the degree, if the allele of disease is located on the non-homologous part of Y chromosome, none of the females should be affected, or every affected son should have an affected father. But according to the pedigree, the son 3-4 is affected and he must inherit YP from his father, who should be also affected, but he is normal, so it's not the case. Also, if the allele of the disease is located of the non-homologous part of X, every affected daughter should have an affected father. But according to the pedigree, daughter 3-1 is affected, should have XP, so you have to give symbols first. So she must inherit the allele of disease XP from the father and one allele from the mother who should be affected. But he is normal, so it's not the case. And If the allele coding for disease is located in the homologous part of X and Y, every affected daughter and son should have an affected father. But in the pedigree, it seems that the disease is not related to the X and Y, so it's autosomal. Calculate the probability for each fetus to be affected by disease, compare these risks, and explain the different obtained results. Does this comparison confirm the hypothesis? First of all, you have to know that 2-1 and 2-2, they have an affected girl, which is the 3-1. So these parents should be carrying the allele of disease, but it was masked. So they should be heterozygous of genotype and P. For 3-2, the risk to be heterozygous, they have family history, so it's 2/3. So 2/3 the risk to be heterozygous for 3-3 and 3-4. So it's going to be 2/3 times 2/3 times the probability to get the allele P or the allele D for a disease, which is 1/2 times 1/2. So the probability or the risk is going to be, this you can simplify 2/2 and 2/2. It's going to be 1/9. And this explains the high risk for this couple due to the presence of the relative mating. Okay, let's move now to exercise 2, talking about the hemochromatosis. So the hemochromatosis disease appears at the age of 40. So this disease is characterized by the accumulation of the iron in the body. It's a recessive disease linked to the HFE gene, which is located on chromosome number 6. So it's autosomal. This gene has two alleles, the normal and the abnormal. The normal allele, which codes for the membrane, that protein that regulates the entry of the iron into the cells, and the mutated one, which codes for abnormal protein that favors the accumulation of iron inside the cell. So here, document 1 presents the partial sequence of nucleotides of two alleles, the normal and the mutated one. Document 2 represent the restriction sites of the enzyme RSA1. So restriction site, it cuts whenever it sees GTAC, it cuts at this position. And this is the nucleotide sequence of both the normal and the affected. So you see here we have GCT, and here we have GCT, GTA, GTA, CCC, CCC, CC, CC, ACG, ACG, TGC, it's here TAC. So here is the difference at this nucleotide at the position 200, 70, 71, 72, 73, and 274, CAG, CAG, and CC. So here the origin of the hemochromatosis according to this is a mutation by substitution at the level of the gene HFE, where a nucleotide G at the position 274 was replaced or substituted by A in the mutated one, and this leads to the abnormal protein. Determine for each of the two alleles the number and the length of restriction fragments obtained after cutting by the restriction enzyme RSA1 enzyme. When they are treated with this enzyme, the normal allele, which represents only one restriction site, GTAC, here we have, this is a cut here, GTAC, and we don't have another this. However, that we have the size will be 241, 2, 3, 4, 5, and here it will be 243, 246 is cut once in T position, 244, and A in position 245, and this will produce two fragments. The first will be about 244 base pair, and the second will be of 387 minus 244, which is about 143 base pair, and the first one will be 244 base pair. However, in the mutated here we have GTAC, so here we have a cut, and also here according to the mutation we have another cut, GTAC. So here we have two restriction sites. Okay, so two restriction sites. So here we have two restriction sites, so we have three fragments. The first one between the nucleotide 243, 246, and the second between nucleotide 272 and 275. So we have three fragments. The first one is the same as 244 base pair, and the second fragment will be of 273 minus 244 will be 29 base pair, and the last one will be about 114 base pair. So here the fragments, here we have 3 fragments in the mutated, and here we have 2 fragments in the normal. The frequency of being heterozygous in this population is 1 over 10. So here the frequency to have to be heterozygous or to be hybrid is 1 over 10. So here to have each of the 2 parents has no family history with a hemochromatosis, so the frequency for each of one to be heterozygous, it's 1 over 10. So being heterozygous, so it's 1 over 10 times 1 over 10. This is for the father to be heterozygous, and for the mother to be heterozygous, times 1 over 2 for the father, times 1 over 2 for the mother. So it's going to be 1 over 400. And this is the risk to have an affected child. A healthy couple older than 40 years belong to this population. This couple would like to know if their children who appear healthy have a risk to develop the disease. That's why they consult a doctor who, as a first step, established for this family a pedigree, which is shown in the document. We calculated the risk, since they gave you the probability to be heterozygous is 1 over 10. So it's 1 over 10 times 1 over 10 for both father and mother to be heterozygous, times 1 over 2 to give the mutated allele, times 1 over 2, so it would be 1 over 400. And as a second step, the doctor performs DNA analysis by applying the Southern blot technique using the restriction enzyme RSA1 and the radioactive probe MP, molecular probe MP, which is complementary to a specific sequence of the HFE gene. This is the MP complementary to a sequence position 273. This probe can fix the whole or to a part of the recognized sequence as shown in document 4. Also, document 5 showed the results obtained by this technique for members of this family. So here we have the technique results. So here it's called the electrophorogram. It shows what? It shows only the fragment to which the radioactive molecular probe it belongs to, or where it is hybridized. So the recognized sequence to which the MP get fixed is only localized at position 273. Thus we have 244 fragment is not hybridized. So this fragment is not hybridized, so it will not appear in the sequence which is hybridized and will not appear also in the electrophorogram. That's why this one is not appear, since it's not hybridized to the molecular probe. Establish the diagnosis for the children of document 5. So you have to establish the children of document 5. As you know that in the electrophorogram we have 3 bands: band 143, band 114, and band 29. So according to this, band 143, it characterized the normal allele. So 143, it's for the normal. And the individual having only one thick band of this, meaning that it's homozygous and is pure. However, the electrophorogram shows bands 29 and 114, they represent the mutated one. So here the fetus 415, it shows what? It shows only one thick band at the level of 143. So this is for the normal and this is for the mutated or the affected. So it will be homozygous and it will be healthy or normal. While 16, it shows 3 bands. So it carries the normal allele and also carries the affected allele. So it's heterozygous, but he is normal. However, that of 17, it shows 2 thick bands in the affected. So this is recessive, homozygous. She will be sick after the age of 40 years. Hence, among the children, all of them, they are normal except for this girl, 17, who will be affected or be sick after the age of 40. Moving to question number 3. Question number 3, talking about sickle cell anemia. Mr. and Mrs. X, they have a daughter suffering from sickle cell anemia. Document 1 is showing the pedigree. This hereditary sickness, whose mode of transmission is autosomal and recessive, is characterized by abnormality in the beta globin molecule, which lead to the deformation of the red blood cells. Mrs. X is pregnant, and the couple demand prenatal diagnosis to know if their second child will be affected by sickle cell or not. So first of all, they said that it's recessive, so you have to give symbols before you give the genotype. So let N capital be the symbol of the normal, and let's say S small for the sickle cell anemia be for the affected. So both parents, they are normal, but they gave birth to the affected child. So the child should inherit the allele for disease from at least one of the parents. So it's mass, and it's recessive with respect to the normal dominant allele. So both of them will be of genotype and S and S. The sick daughter will be S. Based on logical reasoning, what is the probability to have an affected child? Probability of the father to be heterozygous and probability of the mother heterozygous, it's 1. Both of them, they are heterozygous. So it's 1 times father, it's 1 times half to give S and half to give S. So it will be 1 over 4. And this is the probability. Document 2, it reveals what? It reveals the sequence of the part of the non-transcribed. Non-transcribed, it's non-template, it's the coding of the beta globin alleles. HBA who normal, and HBS is the mutant allele of the beta globin gene responsible for the disease and leads to the deformation of the red blood cells. Mrs. X is pregnant, and they demand prenatal diagnosis to know if their child is affected. So here, let's see what will happen here. Here we have two sequences, HBA and HBS. So HBA for the normal and HBS for the sickle cell anemia of the beta globin gene. Here we have two probes, probe 1 and probe 2. Here, HBA, let's see the complementary between them. CTC, and let's see if complementary for the probe number 1, HGAG. So this is correct. CTG, it will be GAC. Very good. Here we have the first 3, second 3. AGG, it will be complementary TCC. Sorry, we have a change in this. So here it is for HBS, and the probe number 2 is for the HBA. It's complementary. So direct diagnostic method by radioactive probe is done for this family. Many copies of the parts of the beta globin gene is can be obtained from the DNA of each person by this technique. These copies, they are separated into 2 lots. Each lot is placed in the presence of different radioactive probe as shown in this. Each radioactive probe is going to bind with either HBA or HBS, and the results are shown in document 4. So here we have the results of probe number 1, probe number 2. As you know that the daughter is affected. So daughter is affected, so she should carry the allele of the disease, which is the HBS. So it has only probe number 1. So according to this, we have mutation at the position 7, as you see here. So mutation by substitution, where we have all of the sequences, they are the same, but they differ in only 1 strand. So the DNA probe 1, it allow us for the mutant, and the probe 2 for the normal one. Thus the probe 1, it allows us to detect that the daughter is affected from the probe number 1. What did they ask you? Specify based on document 2 the location already located at position 7 and its mutation. You have to give the type. Determine in reference to document 2 and 3 which correspond to the probe used. So we said that the normal allele is the probe number 1 for the mutant and probe number 2 for the normal. E. Do the results of document 4 confirm the genotype you have indicated in question A? Yes. Why? Because in the 2 parents, the 2 probes are visualized, as you see, probe 1 and probe 2, which confirm both parents, they are heterozygous of genotype NS. With respect to the daughter, we have only probe 1, so she is affected, while the fetus does not allow us to visualize except the probe 2, which is normal, and it will be homozygous NN and normal. Justify why prenatal diagnosis is more accurate. Prenatal diagnosis is more accurate because it depends more on the gene, and it gives real genotype, and this will allow us to detect the real phenotype. Okay, however, pedigree permit us to detect the phenotype and the possible genotype, not the real genotype. Okay, thank you.